go over, okay, let's just go over the blood supply again. So we're on slide 17. So we're in the kidney and we're coming in, we're gonna come off of the abdominal aorta, and we're gonna go in the renal artery, then we're gonna branch off into segments. We go in between, so interlobular, we go over the arches of the pyramid to arcuate, and then we come out into the cortex, cortical radius. So that's our blood flow. And then we get into the glomerulus, we go through the infiltration thing. We go in the afferent, the big fat one, then we filter the glomerulus, and then we go out the efferent. And then how we get back out is those peritubular capillaries up on top take it out to the blood. And then whatever we don't want, we're going to drain back down the cortical radiant arteries, the cortical radiant veins, the arcuate vein, interlobular vein. It doesn't list it on here, but then the next will be segmental, and then it goes out the renal and peritoneal. That's our blood. We talked about the renal plexus is we're going to change the diameter of those arteries, so we're vasoconstricting and dilating, and we know that that's the sympathetics that always talk to blood vessels. So that's going to help alter our filtration rate. If we vasodilate, lots of flow. If we vasoconstrict, a little bit of flow. And that's going to help make the urine. If we have a lot of flow going in or a little flow going in. And then we got into the nephron, being the structural unit of the kidney. There's over 1 million different nephrons. And here's our renal parts. So we're coming in, renal, artery, segmental, interlobular. We go across the cortical radiate, arcuate up into the cortical radiate, and then we branch off of the cortical radiate. The afferent is the fatter artery, and this tiny arteriole, and it's going to dump it in. The whole thing is called the renal corpuscle, and it's surrounded by the glomerular capsule, which is also called Bowman's capsule. And then inside of it is the network of capillaries. That's called the glomerulus. So filtration is going to occur there. Then we leave, we go out the proximal convoluted tubule. That's where reabsorption takes place. And then we go down the loop of Henle, descending loop, where water goes out, and ascending loop where solids go out. So we're forming the urine in this process. We reabsorb what we want. Now we're going to alter the concentration. Do we want a lot of water? Do we want to hold on to water or let go of water? And over here, do we want to hold on to solids or let go of the solids? Then we have the filtrate. It's in the distal tubule. It's going to look at it, fine tune it. Say, okay, well, what do we need? Do we need to hold on to water or let go of water? Do we need to hold on to solids or let go of solids? And once it's got its final decision, we pass on out the collecting duct and we go out. And from there, we're going out. The collecting ducts are down here in the pyramids, so it's going to drain down through the papilla, the minor calyx, the major calyx, the renal pelvis, down the ureters, into the bladder, and out the urethra. So all of the work is happening here in the cortex and the medulla. The nephron lives in both sections there. And then it's going to drain out and then pass out of the urethra. Questions over the nephron? Okay, so the renal corpuscle, each nephron has one. That's the capsule, the glomerular capsule, or Bowman's capsule is the outside shell. It contains the glomerulus inside, which are the capillaries. They're very porous and they're solute rich and protein free. So protein stays outside. Protein does not come into the kidneys. We need protein to stay outside to maintain that pressure. Remember, pressure pushes out and pressure and things pull in. We get into that osmotic pressure situation again. So proteins are too big to come in and they have to stay out and help maintain pressure. So if we're seeing protein in the urine, that's not a good thing because protein is supposed to stay outside. If you're on the diet like we're talking about, if you're on a low carb, high protein diet, or you're a bodybuilder and you're consuming large amounts of protein, then that makes sense. Otherwise, you're not going to see protein in here because that's going to stay out. So blood comes in through the afferent arteriole into the glomerulus, and then we are calling it filtrate at that point. The fluid is the raw material. that we're using to make urine. Then we go into the PCT, which is attached to the glomerulus, so that's why it's called proximal, and it's going to reabsorb down the lumpopennel. And then DCT, distal convoluted tubule, it's going to be the fine tubular. And all tubules are meandering, and this is so it increases the length of the process. The tubes go back and forth, so it has a longer time to reabsorb in the PCT. It has a long loop down, so water can come out. It has a long loop up, so solids can go out. And then we twist and turn in the distal, so we have time to reabsorb or get rid of. So that's the structure determining the function again, is we want to have time to go through this process, so we make it loopy. It's not just a straight channel from the PCT to the collecting duct. So we have lots of twists and turns to increase the time to allow for these functions. The collecting duct, the DCT, drains the filtrate into the collecting duct, and the filtrate from many nephrons then are going to drain into that one collecting duct. So lots of nephrons are draining into a collecting duct. Each nephron doesn't have its own collecting duct. They all kind of filter into several and then we go into the pyramids. That's why the pyramids have the stripy appearance, because we have all of the loops of Henle dipping down in there, and we have the collecting ducts in there. I know. Stop it. Why do we have to talk so loud?
No, it's the lady down here, there's
Yeah.
a bunch of kids in that room, and she
Yeah.
like yells.
It's like on test day where there was so much noise, because apparently we have to yell at kids to make them hear us. So, very loud going on there. It's hard to concentrate. Up here, I don't know if you guys are hearing it, but we can hear it up too. And so the collecting ducts are giving us that stripy appearance as well as the loop of Henle. And then they're going to drain into the papilla, the pyramids, the papilla, minor, major, renal pelvis. Loop of Henle, as we're going down, the descending limb, that's thinner. If we look at it under the microscope, it's thinner, and that side is only permeable to water. It's going to be made up of simple squamous. Remember, one layer, simple squamous, very thin layer, so water can diffuse nice and easily in and out that layer. Then when you turn the corner, we go up. That section is thicker because we're moving solutes through it. So sodium, potassium, chlorine, whatever it might be, they're going to move up through the ascending loop. Water is not permeable on the ascending. It's staying stuck in the tube there. It's stuck in the loop. It can't get out. And we're going to see different kinds of cells here to allow for solids. So we're going to see like cuboidal and columnar instead of diffusion of water, which would be simple squamous. So different tissues as we're going down through the nephron, because structure determines function, and they're all doing different things. 95% of the nephron is up in the cortex. Only the loop of Henle dips down into the medulla, and then also the collecting duct dips down in there. So here's our cortex and our medulla. Up on top, we have all the action is going up in the cortex. The loop of Henle dips down, then it goes back up, and then the collecting duct. So these are the stripy appearances that we're seeing in the pyramids, because we have lots and lots and lots of tubules going on there, so we see that. Remember, let's blow this picture up. So remember, we're coming across the arcuate artery, then we're going up the cortical radiate, and what branches off of that is the afferent branch. The afferent is bigger, it's going to feed into the glomerulus, and we're going to have lots of filtration going on. It is very convoluted, very twisted, so it takes a long time for it to go through that capillary bed. And arteries, arterioles are, it's coming in, I think, about 85 millimeters of mercury under a fair amount of pressure for a tiny little vessel like that. So we're pushing it in with a lot of pressure, and then we have this smaller efferent artery on the way out. So we have a lot of pressure coming in with a big vessel, and one little tiny one going out. So it's going to back up in that glomerulus, and that's the idea, is we want to have time to be in there so that we can filter it. So it kind of backlogs up into the glomerulus. And then it has that one small efferent arteriole to go out. So it kind of forces the pressure down, and it keeps it staying in there for a while, gives it time to filter. So that's the design of it. And then the efferent comes out, and that's how we get stuff back out into the blood. If it's up on top, we call those the peritubular capillaries. And if it's down in the liquid panel, we call that the vasa recta. So that's how it gets out of that whole nephron and back into the blood, is through those capillary beds. Or maybe we want to keep the stuff in there, then we send it on to the PCT and we reabsorb it, and then we send out 98%, we're going to send back out. How we send it out is through the peritubular capillaries. And then we continue, we let water out, we let solids out, whatever we want to send back out to the body, we send that out through the vasa recta down here. And then finally, we get into the distal tubule, and then urine comes out the collecting duct. So when it's in this whole system over here, this is filtrate. When it comes out the collecting duct, it changes names to urine. The whole process of the nephron is to filter, reabsorb, and get the concentration right. And we're dealing with all kinds of stuff in there, so we call it filtrate. And by the time we've reclaimed everything we want, all we have left is water and the stuff we don't want, and that's what we call urine. And that's what passes out the collecting duct. Questions? So nephrons have two capillary beds. The glomerulus specialized for filtration is unique in that it's fed and drained by arteries. The afferent arteriole is a high resistant vessel going in, has a large opening, so we're getting a lot of fluids in, that's the idea. And then this makes the pressure high, because the more the pressure, the more the flow, right? Remember, flow equals pressure over resistance. So we want a big opening, lots of flow going in, lots of pressure going in. And we need pressure because we're trying to filter it through. And then when we're done with that, we're going to exit out the efferent, and this is a much lower pressure system because now we're forcing it into the tubules, and we don't want to blow the tubules apart. So we go from a high pressure down to a low pressure, and that's part of that filtration flow pressure conversion systems. So we got a lot of pressure coming in, so we're going to have a lot of flow. And then we drop down in pressure, so the flow slows down, and we're going to gently push it through the nephron instead of just slamming it. More of the design. So high blood pressure, we have to have, as in we need to have a lot of pressure coming in. It makes it easy to filter and force fluids in and solids out. So we've got to have a lot of pressure coming in. Flow is determined on that pressure. The greater the flow, the greater pressure. So we got to push it in with a lot of pressure so it can have a lot of force going through that. We're going to drain into the glomerulus, and then 98 to 99% of the filtrate fluid is then reabsorbed in the tubes, and then we return it out to the blood via the peritubular capillaries. The second capillary bed, peritubulars, come off of that efferent as we're going out. So we come in the afferent, and we're exiting the efferent, and that efferent is draining directly into the peritubulars, and then as we get down into the liquid panel, it keeps winding through it.
So, okay, question. So first you're over in the other side, and then you come over to the long
Correct.
part.
Right, collecting them.
And the purple veins are capillaries? Or what is it?
They're up on top, yeah, the capillaries, peritubular around, peritubular around the tubes, or down on the lipopennel, they're called the vasorecta. So yes, capillaries, because that's how we get blood out of one thing into the other. The capillary bed is where exchange all occurs. And so we have the glomerulus is a capillary, and then the vasorecta and the peritubular capillaries are capillaries surrounding the outsides of the tubes. All three of those are capillaries. The second one is peritubular coming off of the efferent. They're going to wrap around the PCT and the DCT. Pressure is much lower so that we have a slow flow. We want to have time to reabsorb things and hold on to things and get rid of things. And it's gonna be very porous because that's what we're trying to do. We're either trying to let things in or let things out. So we wanna have nice big openings in there. The veins erecta, the efferent arterioles don't meander all around. They're much straighter, and so the veins erecta is wrapping around the loop of the tunnel. And they're going to return stuff back out to the blood, water on the descending loop or solids on the ascending loop. So when we're doing that, now we're down in the loop of Pennell, and so we're down in this area, and we're looking at the filtrate that we have, and the loop of Pennell says, okay, do we need to hold on to water or let go of water? So if we hold on to it, it's going to make that fluid more dilute because we have more water, so it's going to be more dilute. If we get rid of it, like we need to keep water in our body, so we're going to dilute that here and there. Or if we get rid of it, it's going to make the water go out, and whatever's in there is going to be more concentrated. So when we're talking about the concentration, is are we holding on to water, making it dilute, or are we getting rid of water, making it concentrated? And then the same thing happens when we go back up the loop, is are we pushing solutes out, are we getting rid of it? That's going to make the urine dilute. Or are we holding on to it? That's going to make the urine concentrated. So when we're talking about the concentration of urine, that's what we're doing, is what are we doing with water and the solids? They can alter it. Hold on to water, dilute it. Push off water, concentrate it. Or we can do it on the opposite side with the solids. We can say, okay, well, we're getting kind of low on sodium, and we need sodium in our system because that runs the whole sodium-potassium pump, and everything operates off of sodium. So maybe we need to hold on to that sodium, and that urine's going to get more concentrated. Or maybe we have too much in our diet, and the body's like, we gotta get rid of that, because it's going to, wherever sodium goes, water goes, and volumes go up, and pressure goes up. So we can regulate it here in the loop, either with the water or with the solids. And then we get it where we want it, we send it up to the DCT, and it's going to take its turn and look at it. So we have a whole bunch of systems put in place so that we don't get out of balance with our solids. And that's our efferent arterioles. The vas erecta is in the loop of Pennell. Either return, that's how we're going to return it back out to it. Okay, questions over that?
The DCT is what again?
The distal
The distal.
convoluted tube. So farther distal as in farther away from the glomerulus. PCT is proximal as in close to the glomerulus. Even though they're right next to each other, it's not that far, but it's not directly connected. So connected, glomerulus, PCT, DCT is connected to the tubule. So it's still, they're still stacked up right next to each other, but it has to go through all this to get to the DCT. Okay, so high vascular resistance, blood flowing through the renal circulation faces high resistance. We have lots of capillaries in there, and remember capillaries are small. So blood pressure drops, it comes in the renal artery, we're coming off of the abdominal aorta into the renal artery, so we're pretty high pressure because we have a pretty large vessel there. And it's coming in at about 95 millimeters of mercury, so that's pretty high pressure. By the time we leave, we're down to 8 millimeters. So we're dropping, and the size is getting smaller, so resistance increases as the vessel diameter gets smaller, that's resistance, and so we're going to see huge changes from where we started and where we end. And that's a resistance feature. Flow, the more resistance there is, the less flow there is, and resistance and pressure opposite as well. So pressure is dropping as resistance is increasing, or pressure is increasing as resistance changes. So that whole formula stays in place throughout the whole system, keeps coming back to haunt us. So what's the summary is the glomerulus is filtering. That's the main idea, filter, filter, filter. The peritubular capillaries are the way we get it out of the nephron and back into the blood. And we're going to reclaim 98 to 99%. That's how we get it out through the peritubular capillaries. Down in the loop of Pennell, we're going to alter the concentration of urine by letting water in or out on the descending, or letting solids in and out on the ascending and it's the vasorecta that returns it to the blood down into the up, higher up, or higher or lower down. That's how we get it back to the blood. All right. This is the one part we didn't look at in lab, and I said write down the letters J, G, A. That's this part right here. So this is still the renal corpuscle. The capsule is Bowman's capsule. This is the glomerulus here. We have our big afferent branch coming in. It's filtered, and it goes out the efferent branch. Or it's going to go down the proximal convoluted tubule. That's the word. So this is what we saw in the last blown up thing on that one model. We're seeing the whole capsule. There's the proximal convoluted tubule. So it's going to go down here. The tubule is going to, PCT is going to look at it, and it's going to reabsorb 98%. On the opposite end, we have this little loop here. That's called the juxta. Juxta means up against or next to. Juxta, what's it up against? The glomerulus. Juxtaglomerular complex is what they call it here, or apparatus is another word for it. And that sits right here, snugged up against the glomerulus, or next to the glomerulus. And it's going to have two parts to it, the macula densa and the granular cells. So that's going to really come into play when we're looking at what are we going to do with this filtrate that's going to go in here. And the juxtaglomerular apparatus is really going to regulate the flow and the pressure of how things are coming in. So how it does it is in our granular cells here, they're lining the afferent artery. Those are going to monitor what's flowing in there, and they're going to vasoconstrict or vasodilate, depending on what they need to do. So they have what we call the mechanoreceptors responding to the pressure in there. And then this part over here, this loopy part, that's the macula densa, and that has chemoreceptors. It's going to have to do with renin, that whole renin, angiotensin, aldosterone. So these two parts of the JGA are really going to affect what's going on, because one's going to vasoconstrict and dilate, mechanoreceptors. The other one's going to monitor what's going to go on, and it's going to regulate with chemoreceptors. ADH, renin, angiotensin, aldosterone. So up here, we're having a lot of regulation going on. Yes? Do you have a question? Okay, so the JGA, juxtaglomerular apparatus, is located near the most distal part of the descending loop. We go down, we come back up, and it's up there on top of the glomerulus. We have two types of cells there that are going to regulate the rate of filtration, and they're also going to regulate blood pressure. So the JG cells are they're large, they're smooth muscle. In the granular cells, this part right here, these are granular cells, and then we have our macular cells. So granular cells are going to secrete that renin. They're the mechanoreceptors. They're going to start the whole effect going on in there, and then they're going to help with vasoconstriction and dilation. The macula densa, so mechanoreceptors, vasoconstricting and dilating in the granular cells. The macula densa is going to be at the top of the ascending loop of Mennel, and it lies next to the granular cells. It has chemoreceptors, so we're going to react to what is going on with salt, sodium chloride, and then it's going to respond appropriately. So one, the granular cells coming in have the mechanoreceptors, and two, the macular cells have chemoreceptors. They're both monitoring what's going on, how are we going to do it, and they're going to handle it in different situations. They're going to monitor the filtrate, and they're going to monitor the blood pressure. They're going to do it in different ways, but they're both doing the same thing.
So that could be a question on the test.
Absolutely.
Like, do they both monitor the blood pressure?
Yeah,
What
like,
monitors
yeah.
and then?
Right. And then, but in different ways. That'd be a good way to put that in there. All right, so looking at the glomerulus, we know it's all about filtration, so how are we doing that? Now remember we looked at, we had the tight junctions, we had fenestrated, and then we had sinusoidal, so we have different levels of permeability. Same thing is going on with our glomerulus, is this is like the blown up picture of what we're looking at. So we have that outer layer that's lining that little vessel, and then we have porous openings so that we can get things in and out. And so we're going to have lots of different features that are allowing that glomerulus to do what it needs to do. You don't have to know all this stuff on this slide. That's just a blown up of well, how are we moving so much through the capillaries, is we have lots of different layers in there, and we have lots of different levels of porousness that allows the water and the solids to move through. So filtration membrane, that's what we're talking about. We have the little capillary, and then it has all these different things. It has 3 layers. It's going to be very porous because that's what we're doing. We're filtering. We want waters and solids to move. So we call that fenestrated. So all the plasma, but not the blood, is passing through there. Proteins are staying outside. It's anchored by that basement membrane, like we saw in tissues a long time ago. And then in that basement membrane, where we have all that fenestrated stuff, in between that and the vessel, it's like a gel-like substance. And then that helps with that electrical activity of all our ions, our sodiums and our potassiums and all that. So that's just going to be another helping factor, is that gel is going to help talk to the ions. And then lastly, remember, visceral is the innermost layer that's actually touching whatever the organ or the vessel is. So visceral, and that has the little feet and slits that are allowing the stuff to go filter down into the capillary. So we're just saying we're filtering in the glomerulus, and how are we filtering? Is we have openings in there, we have a gel that's helping talk to the ions in there, and we have slits in there that's allowing things to move through. So that's just kind of the anatomy of that capillary. And the summary of what we've been talking about, mechanisms of urine formation. So we're bringing blood in, and we're going to adjust that composition. First, the glomerulus is going to filter. Second, the tubules are going to reabsorb it. PCT does the most of it. Lupimetal does some of it, and then the PCT is the fine tuner. And then eventually we're going to go out the tubules, and then eventually we're going to go out the collecting. It's going to have either concentrated urine if we push off a lot of fluids, or it's going to have dilute urine if we hold on to a lot of fluids. But it's the final, the DCT is the final one that looks at the body and says, well, what do we need to do? Push off fluids or hold on to fluids? And then we're going to pass it out the collecting duct as urine, and then it goes down the pyramids, into the papilla, into the minor, into the major, into the renal pelvis, ureters, bladder, and feet. So all of that action that's happening in the nephrons, we call it the functional. That's the whole cycle, is we're trying to get blood in, we're coming in at like 95 millimeters of pressure, 95 millimeters of mercury pressure. Then we're coming in at about 85, so it drops about 10 because the diameter changes a lot. And then we're going to filter, then we're going to reabsorb,